Hi all, I am going to explain about quality control in commercial plant tissue culture. First of all, what is plant tissue culture? Plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of known composition. It is widely used to produce clones of a plant in a method known as micropropagation. Tissue culture is seen as an important technology for developing countries for the production of disease free, high quality planting material and the rapid production of many uniform plants. Then, the quality control. Quality is important in all aspects of tissue culture. The quality of material used will affect the quality of the cultures and the subsequent scientific data and products derived from them. The main areas of quality control that are of concern for tissue culture are the quality of the reagents and materials, the cell line integrity and identification, the avoidance of microbial contamination. Then the reagents and materials. Good quality reagents and materials are available from numerous manufacturers of tissue culture media and supplements. In addition, reputable manufacturers will also carry out a range of quality control tests including screening of mycoplasma and BVDV and supply a certificate of analysis with their products. These state the product and lot numbers and form a vital part of record keeping and tracking of reagents used in the production of cell stocks. It is advisable to further test key reagents such as fetal bovine serum to ensure that they are fit for purpose due to batch variation. Cell line integrity and identification. The sourcing of cell lines can have an important effect on quality. Freshly imported cell lines are a major source of contamination. The advantages of obtaining cell lines from a recognized source such as ECA/CC culture collection are that the cultures will be contaminant free, authenticated by use of short tandem repeat profiling for human cell lines, supplied with a detailed data sheet. Once cell lines have been obtained from a reputable source, it is important to implement master and working cell banking procedures and the associated quality control step such as routine testing for microbial contaminants and confirming the identity of cultures. Then, avoidance of microbial contamination. Potential sources of contamination include other cell lines, laboratory conditions, and staff poorly trained in core areas such as aseptic techniques and good laboratory practice. Thus, the use of cells and reagents of known origin and quality alone is not sufficient to guarantee quality of product, cell stock, or culture products. It is necessary to demonstrate quality throughout the production process and also in the final product. The three main types of microbial contaminants in tissue culture are first is bacteria and fungi, then mycoplasma, and then viruses. First, bacterial and fungal contamination. Bacterial contamination is generally visible to the naked eye and detected by a sudden increase in turbidity and color change of the culture medium as the result of a change in pH. The cell culture may arrive or survive for a short time, but the cells will eventually die. Daily microscopic observation of cultures will ensure early detection of contamination and enable appropriate action to be taken as soon as the first signs of contamination become apparent. In addition, specific tests for the detection of bacteria and fungi should be used as a part of a routine and regular quality control screening procedure. Second one is mycoplasma contamination. Mycoplasmas are the smallest free living self replicating prokaryotes. The effects of mycoplasma infection are more insidious than those of bacteria and fungi, including several long term effects in cell cultures. These can include altered growth rate, morphological changes, chromosome aberrations, alternation in amino acid and nucleic acid metabolism. Mycoplasma contamination is difficult to detect, requiring the use of specialized techniques. ECACC tests cultures for the presence of mycoplasma on a routine basis and offers a mycoplasma testing services. Then third one is viral contamination. Some cell lines contain endogenous viruses and secretes virus particles or express viral antigens on their surface. Example is Epstein-Barr virus (EBV) transformed lines. These cell lines are not considered contaminated. However, bovine serum is a potential source of bovine viral diarrhea virus (BVDV). Contamination and use of infected serum will lead to contamination of cell lines with the virus. Contamination of cell lines with BVDV may cause slight changes in growth rate, but since this virus is non-cytopathic, microscopic and macroscopic changes in the culture will not be detected. What to do in the event of cell culture contamination? One hugely underestimated problem in tissue culture is the routine use of antibiotics. Continuous use of antibiotics is unnecessary and can lead to the development of resistant strains that are difficult to eradicate and may require the use of more exotic antibiotics that may be toxic to cell cultures. In addition, the use of antibiotics may mask a low level of contamination. Once a contamination has been detected, whether it is due to bacteria, fungi, or mycoplasma, the recommended course of action is to discard the culture and continue work with earlier stocks that are known to be free of contaminants or obtain fresh stocks from a recognized source. Aseptic technique and contamination control by personal hygiene. When entering the laboratory, it is important to wash hands since this will remove dry skin and loosely adherent microorganisms which could potentially contaminate cell cultures. Gowns and disposable gloves must be worn. Gloves should be frequently swabbed with 70% of V by V sterile alcohol. Long hair should be tied back to remove obstruction and reduce the risk of contamination. Working within the microbial safety cabinet. When working within the cabinet, the operator should remember that the airflow does not make the environment sterile but keeps it clean. Before any practical procedure is conducted, the cabinet should be stocked with all the materials required for the experiment. In doing so, the operator restricts the number of times that their hand or arm is removed from the cabinet into a non-clean environment. When stocking up the cabinet, it is essential that a cluster-free condition is maintained. All items that enter the cabinet must be sprayed with 70% V by V sterile alcohol to prevent dust and particulates from entering the cabinet. Pipetting and prevention of aerosols. Disposable plastic pipettes 1 ml, 2 ml, 5 ml, 10 ml, and 25 ml are the easiest forms to use for a cell culture. Microbial and cellular contamination can arise through pipetting errors such as spillage of material. Adherence to the following guidance can minimize contamination and safety risks associated with pipetting. Never mouth pipette. Use automatic pipette aids with one pipette aid designated to each cabinet. Ensure the pipette fits into the pipette aid without any force. To avoid contamination, disinfect the pipette aid regularly and ensure that filters are changed regularly. Use plugged pipettes when transferring medium. Avoid drawing liquid into the pipette plug. Use an individual pipette once only. Clean spills that arise immediately with 70% V sterile alcohol. This is the quality assurance of a plant tissue culture. Thank you.